Hi, Dr. Roos here. This is going to be a quick video on the a review for the practical, and mainly this is for students that aren't taking me for my lab, right? Maybe an adjunct's doing your microbiology lab, and I just want to make sure that everybody is getting more or less the same sort of input for what to expect for the practical. All right, because we give the same practical, the adjuncts that teach. Now, I don't know, how when they'll set up the practical, or they may, it's up to them if they want to time, how much you spend at each station, and if they move you around. So, and I usually, even for me, it depends on how big the lab is. If there's like only 12 students or less, then there's no reason to time it and at each station. So you have 90 minutes basically. So I'm going to go through all the different parts of what to expect here. I listed them for the practical, right? So there's 20 stations. There's 100 total questions. Each question is worth 1 point. I'll give you you'll be given a form that's got 50 blanks on one side, 50 blanks on the other, where you put your answers. Be sure you put, if you start at question 40, be sure you put your answer for question 40 under 40. And hopefully, if it's timed, you'll go in the same order of the questions, right, as you move around. So that's another reason to do that. There's 5 questions at each station. Most of the time they ask questions about whatever that station's about. You do have a 90 minutes. After 90 minutes, you'll be asked to turn in your form, and that's been in the past more than enough time. If we do spend 2 minutes at each station, and that takes about 40 minutes for 20 students to go around. Well, if you go to 20 stations, it's 20 minutes, 2 minutes per station would be 40 minutes. That means you're 40 minutes after the hour. So you'd have about another 45 minutes or 50 minutes to go back and answer questions that you left blank or didn't know. If you get to every station in those 2 minutes and you don't know, you leave several blank, 40, 30 blanks at that point, it's a very bad sign that you weren't prepared for the practical. And the purpose of this is to make sure you are. So you'll be given a word bank for, on the bottom of it, it does have a few stipulations, like not all the answers are found. So, you might have 1, 2 answers, A, B, and C aren't there, or colors aren't there, or things like that. So if you're studying for it, use the lab PowerPoint. And then I also now have a PowerPoint that just contains the figures from the Microbiology Lab PowerPoint. And they're unlabeled. And I think that's helpful. So I'm going to talk a little bit about that too, right? So this is the lab practical or lab PowerPoint, and you've used this hopefully to do all your PMP assignments, to study for quizzes for the lab. One of the stations on the practical will be a microscope similar to this. It may not be exactly like this because we got new microscopes, but the ocular lens, you need to know all these. The fine focus knob is over here, right? Light source or lamp is down here. The stage is the bottom here, right? You should know the objective lenses, right? You put your slide on the stage, but there's not much to that. And then you might get a question or two about compound microscope, what that is, what is parfocal. You might see the definition. Like I think on one of my quizzes, I had the definition of resolving power, the working distance. So be familiar with those terms, what they mean. So if you get a question, one of these might be the correct answer. When you heat fix down here, right, a smear, it's to attach the bacteria to a slide. there's not going to be a station about the smear, but you might get a question. I think one station is just questions without any figures, right? So I might ask you, about heat fixing, okay? And then the, when you get to each station, most of them, you'll get an image, say, like from the PowerPoint, like this one here, right? And if you see this, then you need to, the first question you probably get asked is, what is the name of this stain? You need to tell me. Now, what I did with these figures, the PowerPoint figures, is this is unlabeled. So you can go through this and test yourself, right? What stain is this in case this picture is used, right? What's the name of this test? Now, if you don't know the name of the media when you're taking this, then the 5 questions that ask about it, you can't get correct. That's the issue that many people have. And the other thing that I advise students is just because you can't answer the first question, don't move on to, you need to read the second and the third and the fourth question. Those questions may tell you, or help you figure out what station it is. So read all the questions. Don't go, I don't know the answer to the first question, and then don't move on. And so I think this will be helpful having this. Now, this one is for the methyl red test, right? But the MRVP media is what's used for this. So if I ask, what's the name of the media used for this test, it would be the MRVP media. Okay. So anyway, I think that one will be helpful. The gram stain, right, will be one color. I tell students there won't be 2 colors. Whereas if you do the acid fast, if it's a microscope or if it's this image, you can tell the difference because on the acid fast you have pink and this blue color, where on a gram stain there won't be 2 different colors. So if I ask about the green objects and you see a slide similar to this, or if it's the microscope, sometimes there's a microscope. I've got slides, that'll have the spores on it. But if one of the questions out of the 5 say, what are the green objects that you're viewing, then that tells you it's this stain, right? There's no other stains with green objects. So keep that in mind. All right, this is the capsule stain, right? And these are the questions will be asked about primary stain, decolorizer, or what is the white halo? Could be capsule, or I say, what is the structure? I won't say capsule, I might just use the term, what is the color of the structure as a result of this stain? That kind of thing, right? What are the purple objects inside the halo, right? They are the bacteria in this case. So be able to recognize this, and right, these are the media. Pink, green sheen, hopefully, this is a dead giveaway, right? It's the EMB. The other thing that you need to keep in mind is the question, usually the first question, not always, but usually the first question is, what's the name of this media? EMB. What's the name of the inhibitor? ESM-ethylene blue. What's the name of the indicator? ESM-ethylene blue. So you can end up with 3 questions having all 3 answers, or 2 questions having the same answer. Don't think that you check it off the word bank and you don't, you can still, use the same answer. Gel tube, starch plate, be able to recognize this. Which one's positive, A or B? I might ask that kind of question. There's not a station for the blood agar plate, but there could be questions about the blood agar plate, what's the inhibitor or indicator for it, and descriptions of these types of hemolysis. So be sure you know that for it. All right, this is the phenyl red broth, right? The Durham tube is what has the bubble. Citrate agar, this is pretty easy. And then to tell that you're at the methyl red test or the Vogts Proskauer test is the terms. If you see a question about neutral end products, Vogts Proskauer. If you see a question referring to what mixed acids are generated, then it's the methyl red test. That's how you can differentiate these two. They both use MRVP media. The name of the test is methyl red test. The name of the indicator is methyl red. So, keep that in mind. Right? Acetoin is generated sometimes during glucose fermentation. And then so if methyl red, if this test is negative, this one might be positive. Right? You use VPA, VPB, you use the MRVP media to complete this test. Right? And we're referring to this test because you get a question about neutral end products. And then the sim media, this one's pretty complicated. Be sure you recognize it. Same with this. Be able to recognize this, right? TSA plates are used. Some people get these two confused, oxidase and the catalase test. Okay, you use a TSA plate. You just, you can put the bacteria on anything really, and then add oxidase to do the test, or add peroxide to do this test. So, and then this one's pretty straightforward. Kirby-Bauer, what generates zone of inhibition? Antibiotic does. Or, right, what's the area around the disc called? That's where the bacteria don't grow. Zone of inhibition. Okay, and there's no questions about this one, so I just wanted to do a quick video, and be it a review for the practical, so that, like I say, the students that aren't in my lab, I usually go over all this, and so anyway, good luck on the practical, right? And I'll once the practical is graded, and then I'll put all what you'll have total lab points, and then I will put them into the grade book for the class. And then sometimes there might be some bonus points for lab attendance. So in that case, I'll add them to your lab points if I do that.